Thank you. I bring greetings from the Indian Association of Medical Microbiologists, Pondicherry chapter. It is immense pleasure to come and participate in AME on AMR pandemic, Mirage to PHAGE therapy. And I would like to congratulate Dr. Vinod and Dr. Gerald for organizing this CME at a very short notice. As we are all aware that this AMR pandemic, which is a silent pandemic, which is going on, and we have millions of deaths which occurs because of this antimicrobial resistance. When we sit every day for reporting, and when we start reporting and Acinetobacter baumannii or Klebsiella pneumoniae resistant to all, nowadays including resistant to colistin, then when the clinician comes and asks, what next, ma'am? What are we supposed to give? And then we are in a limbo, what to tell them? And including even Enterococcus faecalis, we are getting a lot of cases of vancomycin resistance. We need alternatives. There are drugs which are there in the pipeline, but then again, we don't get this new compound which are there. So we have to find alternatives. And one such good alternative is this PHAGE therapy. And I hope all the participants would benefit from this. And you youngsters should take up research on this. And so we can get results which will help the mankind. And I wish this academic feast would be very fruitful to everyone. I would request all of you to actively participate. You have a good lineup of all the speakers who are well versed in their own field. And I think we should make best use of this. Thank you. And thank you for inviting me.
Thank you. We'll proceed with the third session. The topic would be biofilm be gone, bacteriophages to the rescue. The moderator will be Dr. Sheila. She is Professor of Microbiology from PIMS. Madam is Secretary IAMM. Her field of interest are AMR among enterobacterials, healthcare associated infections, prevention and control. We welcome Sheila ma'am to moderate the session.
Thank you, Dr. Gerald. So today's the third session is on biofilm be gone, bacteriophage to the rescue. And the speaker is Dr. HIREN JOSHI. Dr. HIREN JOSHI is a staff scientist at Baba Atomic Research Center, focusing on bacteriophage-based biofilm control. He's developing India's own phage bank for the therapeutic applications. Notably, his contribution is that his nitric oxide releasing own dressing for diabetic foot ulcers has completed phase 3 clinical trial and received DCGI clearance for commercial launch. We also have a panelist, Ms. Krupa Parmar, who is a microbiologist and phage researcher at Precision Biotech Therapeutics, USA. She designs and implements clinical trials protocols for lysine and phage therapy application, aligning with ethical and scientific standards to achieve regulatory approvals. So I think Ms. Kripa is there.
Hi, can you hear me?
Yes. Yeah, I think we will now have Dr. HIREN to start this talk. And as per the previous sessions, if you have any questions, please put it up in your chat box. Then we can have the discussion at the end of the presentation. Thank you.
So the question comes, and I think so few people asked this question during this Gopanath sir's presentation, that yes, okay, we understand phage is great, phage is doing well, but from where we get the phage? And believe me, the phages are omnivorous, and the country like India, which we have such a diverse, ecosystem. You name the bacteria, and you will get it in your sewage sample. And if you get that bacteria in your sewage sample, you will get phage from the same sewage sample, because it is an obligate parasite, right? So wherever there is a bacteria, there is a very high chance you will get the phage. So getting the phage from the environment, it's not that big challenge, and particularly a country like India. And that's why I always say that we should take a lead in a phage therapy, because the kind of phage we get in India, you will not get. I give an example of Scotland. They want to work with the Vibrio cholerae phage. but they could not get a single phage against Vibrio cholerae from Scotland because there is no cholera. But if you come to the West Bengal or Bangladesh or that border, you have extensive cholera outbreaks. You get n number of phages specific to Vibrio cholerae. So that dividend we should take. And there is a time if we come together, India can be the supplier of phage entire world. Presently, which is dominated by the country like Georgia and the old USSR countries. But India have that advantage that we have a different kind of phage, we have a different kind of bacteria, and whatever you want, we get it. I'll give you one example also, which Atif has done. So basically what I meant to say that the getting phage is not a very challenging task, right? So we are getting phage across various systems. And as I mentioned, we were working with the bacteriophage since last 5 to 7 years, particularly for biofilms control in the cooling water system and all. And as a spin-off, when we started thinking, why can't we use whatever the experience we gain in isolating the bacteriophages with different environmental sample. Okay. So we started developing a bacteriophage collection in our system. And because we were working on the phage, we have an entire ecosystem or entire setup which is required to, from isolation to purification, doing the whole genome of the phage and making it ready for the use. So with that setup, we start building what we last 2 to 3 years, we started building what we call as India's own bacteriophage or a phage bank. So the idea is we should have a phage collection from entire gamut of escape pathogens and the most prevalent pathogens which we seen around the hospitals. And whenever patient comes which have a multi-drug resistance and there is no option, we should able to give phage immediately within 2 to 3 days. So with this goal in mind, we started building a phage bank. And I'm happy to share that we have almost now more than 300 phages specific to all the WHO or ICMR priority pathogens with us. And the collection is keep building. So every day we are adding few phages up and down, right? So in this particular context, again, I want to come back to all my friends or the IDs and doctors who are participating in this small workshop or a conference. What we need is now we have a phage, but we want to test it against the clinical isolates. More the screening we do against the clinical isolate, more the robust phage cocktail we can prepare. And that's why if we come together again, please give us a clinical isolate which you find in your laboratory or in your microbiology lab every day, which has a pan antibiotic resistance or very nasty kind of the organism, please send it to us. Maybe Dr. Gerard can coordinate. We will run our phage, and with the experience, we run again and again. And end of the day, if we run, let's say we are targeting Pseudomonas, if I run my phage against 150 isolates of Pseudomonas, what are the phage cocktail I will get, which is effective against all 150 Pseudomonas, then I will be quite confident that whatever patient comes, you give this phage, it will work. So more the screening, more the isolate, more the screening, we get a better and better phage cocktail. And that is the idea where you can collaborate with us, you just need to send us the strain, rest we will take care. So I just give the slide. This is how the entire process we have developed in our laboratory. For example, the first start with the finding the phage. So as soon as you get the isolate, we'll test, or we take the environmental sample, whether it can be sewage sample, it can be river sample, it can be a hospital effluent treatment plant or anything. We see whether the phages are there or not. Once we get the phage, what we'll do, first we test it against the particular organism, whether they are sensitive or not. Once we do that, then we do the whole genome sequence of phage. We use the nanopore platform to do that. This is mandatory as per the requirement, a regulatory requirement, that your phage should not have any antibiotic resistant gene, it should not have any virulence gene, and it should not be lysogenic. It should be lytic. So this verification we do with the whole genome sequencing. Then if required, there is an in-between stage where we use some infection models like G. mellonella, where its innate immune systems are very close to human or mammals. So if you want to test whether like you infect them with the Klebsiella, then inject your Klebsiella phage and see how they survive and all, which gives you a fair idea that how the bacteriophages are effective inside the body. And once that is done, we have a phage then we go for a production. So production, we can go up to 5 liter scale, and then we use a TFF system, which is called tangential flow system, to concentrate the phage. So we concentrate from 5 liter to maybe 10 ml, and we determine what is the titer. So once you pass through the tangential flow system, it takes care of all the endotoxins and other impurities. So you have a pure phage available with you, which have a titer of, let's say, 10^11 or 10^12, because you are concentrating it. And then you can make a dose as per the requirement. As Saroj mentioned, the ideal dose is around 10^7 to 10^9 phage, which can be put into the saline, and you can either, if the wound bed, means directly spray onto the wound, and very simple application. You can simply spray. You can have a cotton gauze, sterile cotton gauze, dip into the phage suspension, put onto the wound bed, it will work. So you don't need any specific what we can say preparation for per se. And in between, we do have some quality control check that what are the phage we are giving are pure or not, or whether anything is there. So there are some QC parameters which we take care of it. So what in summarize I'll say is that we get the phage, isolate the phage, to finally we can prepare the phage formulation and give it to you to put it onto the patients. So that entire systems is quite established protocol in our laboratory where we do this. So again, coming back to the same table, we have now more than around 300 phages against various pathogenic organisms, and we are primarily focusing on the escape and the WHO ICMR prior to pathogens. So this is the one case which I was mentioning. Actually, there was one patient in Switzerland suffering from cystic fibrosis. And they also have their own phage bank, and they got it around almost 154 phages they have screened, and none of the phage was working with that Pseudomonas. So there is a one platform we called as a phage directory where people interact, all the phage researchers interact, and there this request has come whether anybody have phage against this particular pathogens. So they have sent us the strain from the Switzerland, and we got the phage, which was quite lytic on this particular pathogens, and the phage preparation was sent to Switzerland for the treatment. So again, I coming back to what I was telling is the dividend, the diversity and the advantage of India. The phage they tried all the phages, none of them was working. But we got, I think, in the first shot, and Atif is here is I always say, is a phage hunter. You give any organism and he will get you phage within 1 week. So he got it within 1 week and the phage was sent. So that is the advantage of India. You name it and you have it. Right. So that's what we should take. And going to little bit of history, why the phage therapy has come back to the limelight again. This is the classical case of Professor Steffin. So her husband got infected with multi-antibiotic resistant acetonobumani, and she tried a lot, nothing was working, and I think doctor almost say that nothing can be done, he has to go. But then she met some phage researchers, and the phage formulation was developed by Texas A&M University, and he was completely cured. And now she's become one of the most prominent advocate of phage therapy in the world. Same way like Pranav Johri, who is going to give a talk here, he is the person who got benefited from the phage therapy. His life was saved because of phage therapy, and he is now propagating this phage therapy in India. And if you see the historically, it is not new, as Gopalnath sir was telling that it is 1928 onwards, people are working with the phage. There were lots of phage preparation was developed, cocktail was developed, and extensively used across various places. And most importantly, is in a cholera and a plague outbreak in Punjab. So that was the first time the phage was used to control cholera, and it was significantly controlled using phage. They have given to the patient, as well as the smart guy knows that the cholera is due to waterborne disease. What they have done, they have injected phage into the well of that particular Punjab community, and the cholera outbreak was controlled. So there are thousands and thousands of success story of phage therapy. We are not going to do anything new. It is already there in history. Unfortunately, during World War II, when the penicillin came into the market and the industrial scale production of antibiotics started, we lost the interest in the phage, because phage are very specific. And that's why the phage therapy was gone bad by almost 50 years. Nobody was working except those USSR countries. But now again, it is coming in prominence. And just for your information, I'm saying, for example, India is the largest exporter of aquaculture products, particularly prawns and all. And as per the guideline, even 1 ppm of antibiotics detected in those prawn culture or something, the entire consignment get rejected. That is the international law, right? That's why there is no antibiotics is used in aquaculture. Same way with a certain poultry which we are exporting. And now in India, particularly the company called Salem Microbes in Salem, our own Tamil Nadu, is producing a phage cocktail against Vibrio, Vibrio harvey, which is the main infection causing agent in the aquaculture. And that is deployed at 40 lakh liter capacity. So if you see the aquaculture plant, 100 ml of phage formulation, just add into that and you take care of Vibrio. So there are success story at the large scale, at the smaller scale, at the laboratory scale, everywhere. We just need to embrace this new technology with open heart and take it to the next level. So as I mentioned, it was extensively used, across. I also want to take this opportunity because lots of infection control people are here in this particular talk. We always talk about AMR in terms of excessive use or abuse of antibiotics. But one factor we always forget or we don't give much emphasis is the biocide problems. See, we use extensively a biocides like BKC, CPC, all the disinfecting agent which you use in your ICU, you use in everywhere in the hospital. They are becoming a major problem because there is a strong evidence now, even in our own research, that biocide also leads to cross-resistivity to antibiotics. And that is predominantly governed by what we call as an efflux pump. So I want you to just take care of this part also, that when you use the disinfecting agent or a biocide in your hospitals or various application, please be judicious with it. Because once the bacteria become resistant to those biocides, they also become resistant to antibiotics, and that is primarily governed by the efflux pumps. Right. So this is what we got because in our power plant also we use lots of chlorine and chlorine dioxide, those kind of biocides to control the biofilms. And we observed that, for example, we isolated one organism, Klebsiella pneumoniae. It is very related to Klebsiella pneumoniae. And it was, kind of resistant against all antibiotics, all the biocides, what we use. And we also found that they are also what we can say resistance to antibiotics. And that was a problem. None of the biocide works, no the antibiotics work. So again, we have to go back to the phages to control this. As I mentioned, we use a nanopore sequencing platform. So when we sequence this organism, Klebsiella pneumoniae, if you see here, it has a OKP family of antibiotic resistant gene. But if you see the efflux pump, it has a hell amount of area of efflux pump. Efflux pump is nothing but as soon as any molecule goes into the cell, it will just push it out. And when the organism have this efflux pump, they become resistant to, because if you see antibiotic resistance, you have a particular gene, let's say beta-lactamase. That gene only imparts the antibiotic resistance against beta-lactam. But when you have efflux pump, many a time those efflux are very broad spectrum. It means it gives the resistance against, let's say, beta-lactam, it can give to tetracycline, it gives to not all. So even biocides. So for that, why the efflux pumps is very, very critical, and we should focus on efflux pump when we talk about antibiotic resistance and how it grows. And what we observed is because of excessive use of biocides or disinfecting agents, which leads to development of this resistance organism, which have the efflux pumps and which also provide a cross-resistivity against the antibiotics. So we should take care of it. So this is a small essay which we call as ATBR efflux pump, and you can see that in the last CPC and BZK, the entire ethylene bromide which is going to the cell is going, going ejected out of the cell because of the high flux pump activity. So this is very, what I can say, a deadly combinations. So for example, when we use AMR, we talk about AMR, we say we should use, biocides to control the AMR. We should sanitize the surface. We should make sure that it should not spread between this thing. But sometimes the same biocides or disinfecting agent will impart the antibiotic resistance to organism. And that's why we should really think about use of biocides or disinfecting agent in our setup. So we have isolated phages against the Klebsiella pneumoniae and Enterobacter cloci, and we have shown that though it is highly resistant to antibiotics as well as biocides, we can effectively control it without any problem, even in the biofilms. And interestingly, this organism was enhancing the biofilm formation in presence of antibiotics and in the presence of biocide. So it became a double problem. You have a biofilm acid problem, When you use this agent, it enhances the biofilm formation. So it becomes very, very difficult to treat unless you use the PHAGE. It's very difficult to treat this kind of organisms. So another bacteria we got again from our system is Enterobacter cloci. Again, it's a opportunistic pathogen. And same story, efflux pump, resistance to all antibiotics, resistance to biocides, and what we use again the PHAGE to control this particular organism. And this is the one small slide, the very important, I think everybody talks about the advantage of PHAGE. Though the disadvantage, you need a personalized PHAGE therapy, but the advantage is you can save the microbiome, what Dr. Gerard also mentioned in morning. And as we know, last 10 years is a boom in human gut microbiome. And now we know that the microbiome of human or gut microbiome can affect from your mood to your intellect, your IQ, to autoimmune disease, and so forth. Right? So you need some therapy, which is quite narrow spectrum. But for that, we need a collection of phages which can immediately deploy. You don't need to wait till personalized phage is isolated. And that's what we are trying to build. So I'll also take this opportunity to mention whatever one of our inventions. See, nitric oxide is again a bioactive molecule, right? So we were using it for biofilm control in our system. And as a spin-off, we thought, why not NO can be used for wound healing? So as you know, the nitric oxide, this is nothing to do with PHAGE. It is just I'm mentioning if anybody who is interested in diabetic foot ulcer and want some new alternative for the treatment, I'm mentioning from them, because this is November 2024, we completed phase 3, and now it has got approved by CDCO for commercialization. So by maybe this month, we will be commercially launching in collaboration with our technology partner, Collagenesis Pharmaceuticals, again from the Salem in Tamil Nadu, because they have expertise in collagen. So the advantage of this particular wound dressing is it has a nitric oxide, which is a potent antimicrobial agent, as well as it has a very active role in wound healing process. So in the diabetic patient, what happened is because of the polymicrobial infection and the high blood sugar, it don't get healed. The infection control is very difficult. In addition, because there is a NO synthesis pathway in the diabetic patient gets impaired. So they don't produce nitric oxide, which is required for healing like a normal person. And that's why this dressing provides this both. It gives the nitric oxide from the outside. So first, it takes care of polymicrobial infection, and it also enhances the wound healing by because NO is known to have a angiogenesis property. It induces the capillary formation. So any necrosis or anything is there which get the vascularized very quickly and become granulated, blood flow increase. Even neuropathy gets eliminated within the one dressing change. And it's a very fantastic product which is coming up. So please, anybody diabetics food specialist, if you want, they can use this product. Maybe it's launched in the next month. And this is the name of the product. It is called Colanox. It is a collagen-based hydrogel product. And you need to just rehydrate before applying onto the wound bed, and it's start taking care of the chronic wounds. So with this, I would like to conclude. And as we I always say that though Department of Atomic Energy has a mandate of doing lots of work towards the power production and the national security, but we also there for the society. We do lots of work, and we need your support to whatever we have in our lab reach to the right users in the society. With this, thank you so much for opportunity. Thank you, Gerard. Thank you, sir. Thank you so much.
Thank you, Dr. Joshi. I don't think any questions are there on the chat box. Do you all know online? If there is any questions, can you all just post? Maybe we can wait for a minute.
One question has come up.
Yeah.
Do biocides also affect bacteriophages?
Do biocides also affect bacteriophage? Is the question, sir?
Yeah, this is a very interesting question. And fortunately, the bacteriophages have much more resistance to biocides compared to the bacteria. And whatever the bacteriophages we have used for against the Klebsiella quasi-pneumoniae and the cloci, all the bacteriophages are biocide resistant. Anyway, antibiotic does not affect them, but biocide do affect. But whatever the bacteriophages we got, they are highly resistant to biocide also. So you can use along with the biocides.
Actually, I have a question because generally when we see bacteria, means the biofilms, we have problems with all this prosthetic prosthesis which is being put, or even just an IV line, the central line catheters which are put. Most of them actually get colonized with Goggulus negative staphylococcus, which finally end up forming biofilms and all that. So, do you have any phages for this Goggulus negative staphylococcus?
Staphylococcus, we have few phages, but as of now, we have not worked much with the staphylococcus. But I think this is interesting inputs. We will definitely get the phage, and particularly we get the phage which have more the depolymerase activity, so you can reduce the biofilms.
Thank you. If there are no more questions, one more question is there, I think.
Bacteriophage spray on hospital environment.
Yeah, in fact, even I was thinking about it. Bacteriophage spray in hospital environments for the disinfectants.
Yes, again, it's actually I forgot to mention that why I mentioned. Yes, bacteriophage can be used as a spray, and we did work with the Pseudomonas. Particularly, if you remember, last year there was an eye infection report from the US, and the primary reason being the Pseudomonas aeruginosa and the water which was there in the eye drops, which is manufactured in near the Chennai. So we were thinking on that line, can we, because Pseudomonas is a nasty organism in a water disinfection as well as the surface. So we have developed a formulation, and the good part of this formulation is it's stable at a room temperature up to 6 months. You no need to keep it at the refrigerator. The phage formulation is stable at a room temperature, so you can use as a spray wherever you want a surface. And that's what even we suggest, like for example, you have some catheters, or you have some tools which you use for operation or something, can be treated with the phage. So it can be, take care of any biofilms or any infection present there.
Phages for anaerobic bacteria.
Okay, there's a question on phages for anaerobic bacteria.
Yes, there are bacteria. We can get bacteriophages against the anaerobic bacteria. In fact, we were trying to get with SRB, sulfate-reducing bacteria, which is a problem in our system. But it is little challenging in terms of, because most of the anaerobic bacteria, either growth is less, or see, the main technique of isolating phage is the plate-based method, like you plaque formation and then gets. So unless you have a very good anaerobic chamber where you can put your plates and prepare the media and all, it's difficult to isolate anaerobic, bacteriophages against anaerobic bacteria. But yes, they are there, and they are definitely helps in eradicating the anaerobic bacteria.
One Health phage.
Yes.
We're asking this question.
Yes, it's in fact the recent what we can say, there was two hearings in European Union parliaments about the phage and the One Health concept. See, One Health concept says that everybody should be healthy, right? Your environment should be healthy, yourself should be healthy, your economy also should be healthy. So the example which I gave about the aquaculture, where they have completely eliminated the use of antibiotics, particularly those who are for export. Same way with the poultry. For example, Proteon Bio have opened their facility in Nashik recently. They are developing a phage at the commercial scale for a poultry application, particularly Salmonella. So what they do is they just feed this phage into the water channels where the poultry, means the chicks, drinks the water. And they able to completely control the Salmonella growth. They no need to give any antibiotics. So yes, phage has a very critical role in One Health concept, where you can minimize the antibiotics usage across all the domain, and which is quite extensively. I think morning also, sir was telling we should try first with the animals and all. But that is already done. There are almost 10 to 15 companies which are commercialized the phage formulation for the poultry, for the vegetables, for the aquaculture. So they are already in the market. And yes, it is a very important part of One Health concept.
Phages dental caries. Phages dental caries.
Dental
Okay,
care.
this One Health concept, even like if you see, compared to the antibiotics which are being used in our medical profession, If you look at the use in agriculture, the poultry, it is like in aquaculture, the amount which is used is huge when compared to actually what the medical care where we use it. Okay, the next question was on dental caries. PHAGE in dental caries.
It can be used provided we know the organisms present, a predominant one. In fact, as far as I remember, there is one company who is developing mouthwash using the bacteriophages. So like your chlorhexidine or something like that, they are using it to develop, using some predominant strain present on the dental and all. They are developing the mouthwash with that. So yes, there is a possibility, provided you know the composition of the bacteria.
I think how to use biocides judiciously as they may spread cross resistance in bacteria. That's a question.
It is little difficult to implement per se because you need a biocide or you need a disinfecting agent in your system. So the best way of doing is the rotation. And I think many hospitals follow that, particularly the infection control department, particularly the hand hygiene solution you use. You have a rotation. You use one, then the next composition. So same way the biocide also we need to use it in a rotation which can takes care of this particular part. And again, as the PHAGE I am advocating, so we can also try to see that because most of in the hospital or any setup, you need to take care of only 2-3 nasty organism, which is prevalent. You don't need everything to die. So maybe you can offer a PHAGE formulation which can particularly takes care. And the another advantage of PHAGE, which I can say, particularly the sink and the water where people use. See, most of the infection in the hospital, people have found out finally comes from the sink where people wash their hand and all. So once you dose the PHAGE, you don't need to dose, repeatedly because they are auto dosing. If there is a host, they multiply, increase in number. So you no need to do every day. You do once or twice, it takes care of it. So that is another advantage of PHAGEs compared to other disinfectant.
Thank you, sir, for that excellent presentation. I think, at least for me, there are many new informations which I have learned from this. I hope it was the same with all the participants. Thank you so much for that wonderful presentation. I think we'll move on to the next.
Thank you.